Okay, we'll move on to our next speaker who needs no introduction. Dr. David Livingston is well known to all of you, and David is going to talk to us about ERBB3 as a new target. David.
Thanks very much, and I thank the organizers warmly for the privilege of coming to talk to you today. As some of you might know, our group at Dana-Farber has for years been spending considerable time on trying to unveil what are too many secrets to count regarding how it is that two very powerful germline alleles, BRCA1 and 2, suppress the development of breast and ovarian cancer. And during that period, began to develop an interest in the way in which ovarian cancer develops pathogenetically. And in the course of doing that, I fortunately met two very smart, absolutely brilliant young people. If we could try the first slide here. It's self-evident. So is this. Two young people. This pointer here might help. Joyce Liu and Qing Sheng. Joyce Liu is a physician scientist, a trained oncologist at Dana-Farber whose interest is in the treatment of ovarian cancer. And Qing Sheng is a basic scientist. And these two people formed an incredible collaboration in the group and began to explore a series of phenomena that have resulted in data that's recently published. I should also point out that, if we could go back one, please, that this work is a product of extended collaboration among a family of investigators, including my dear friend and former postdoctoral fellow Ronnie Drapkin, also at Dana-Farber, Bill Hahn, Andy Kung, Matt Meyerson, and Tom Roberts from Dana-Farber, and a very hearty and highly intelligent group of colleagues at Novartis and other colleagues at Merrimack Pharmaceuticals. So the story begins with a screen, a simple screen carried out in the early part of this decade, in around 2006, by Qing, with a pipette, no robots, nothing, in collaboration with Bill Hahn, who very generously led us at that time, a burgeoning group of hairpins, n equals 3 to 4 each directed at a different tyrosine kinase. The question was really a lark at the time. Were any ovarian cancer cell lines, it was hard to obtain primary replicating hearty ovarian cancer cells at the time, were any tyrosine kinases capable, after depletion from such cells, of eliciting a therapeutic-like anti-proliferative, some sort of nasty phenotype. And in a number of experiments, this is one here, which were basically repeat screens done with 3 different non-primary established cell lines, ERBB3, KDR, and FA4 scored as positive. But ERBB3 almost always scored as positive with 3 out of these 4 hairpins, and with 3 out of 4 hairpins that were non-overlapping to them, and so on and so forth. And we decided, therefore, to pursue this observation and analyze 17 different established human ovarian cancer cell lines, Joyce and Qing did, and made the following observations. Some of these cells, most of them in fact, synthesize ERBB3, which, as you know, is the stepchild of the HER2 family, or the ERBB family, stepchild because it's not a kinase. It forms heterodimeric partnerships with the other 3 members of the family, the EGF receptor, HER2 and HER4, but it itself can't be a kinase, and it acquires, through transphosphorylation, in part delivered by its tyrosine kinase heterodimeric ERBB family partner, a tyrosine onto any of 12 different signal transduction sites on the surface of ERBB3. And when that happens, ERBB3 becomes phosphorylated here on position 1289. And if ERBB3 signals in a traditional way, as we knew at that time, it was capable of forming a non-covalent complex with a regulatory subunit, P85, of PI3 kinase. And this led to activation of AKT, the turning on of the PI pathway, proliferation, activation, and so forth. So the sum of this experiment in these colored arrows here is that these 17 different established ovarian cancer cell lines, each from a different patient, most of them papillary serous in origin, came in 3 flavors. There were some characters like H8A that produced no ERBB3 at all. There were some characters in the red that produced ERBB3 and activated it, and in most cases, the activated ERBB3 was associated with the regulatory subunit of PI3 and therefore was engaged in PI3 activation, or so one could presume, albeit on relatively limited data. And then there were some guys in blue that produced ERBB3, but it was inactive. And I draw your attention to this cell line called OV5, or OVCAR5, because we used it as a non-isogenic control for this cell line called OV8, which not only produces ERBB3 in substantial amounts, it activates it, and the activated protein interacts with P85. That's not the case in OV5, which produces almost as much or as much ERBB3 and doesn't activate it, and the protein doesn't interact with P85. So if you take OV8 cells, as Ching did at the time in collaboration with a group at Novartis, and generate inducible hairpins, 3 different ones, non-overlapping, each against ERBB3 and a control, you can see that for each of the 3 non-overlapping ERBB3-directed hairpins, an anti-proliferative response was generated in these stable, non-clonal mixtures of stably transformed cells. When you add dox, that is, you turn the hairpin on, and not when you don't, in blue. Compared to control, turn it on or not, the control has no ability to attack ERBB3, and so this suggested that ERBB3 synthesis in this cell was correlated with the ability to proliferate, correlated with. Now, if you do another control and take those OV5 cells, they produce plenty of ERBB3, but they don't activate it, and do the same experiment in them, you can deplete the cell of ERBB3 when you add dox, 3 different hairpins, but there's no anti-proliferative response in keeping with the specificity or the proposed or alleged specificity of the experiment. Still a correlation. The presence of ERBB3 and its activation is correlated with the ability to sustain proliferation. Doesn't say it's absolutely required. So the next question was, how about other cell lines? Here's a line, iGrove1, another one, SCOV3. They have the same molecular phenotypes as OV5 and OV8. Attack this character with a hairpin directed against ERBB3 versus this one. You get an anti-proliferative response here, not here. There's plenty of ERBB3 in both, only this one activates it. So the previous results are not epiphenomenal. And then you can go further and ask, okay, this is the era of hairpin screening in which off-target effects are legion. And so Joyce and Ching did this experiment using siRNA, which was easier to manipulate in transient transfection experiments. They drove down the concentration of ERBB3 here in the green in these OV8 cells, obtained an anti-proliferative response, and could reverse it partially, only partially, by overexpressing with a wild-type allele of ERBB3, an ERBB3 CDNA. But when you add neuregulin 1 alpha, in fact, neuregulin 1 beta in this case, the natural ligand for ERBB3, which is capable of promoting heterodimerization between ERBB3 and another partner, now we see almost or complete reversal of the negative phenotype, in keeping with the notion that the negative phenotype obtained by dropping the concentration of ERBB3 in cells is not the result of an off-target effect, at least in tissue culture. Further supporting the notion that there is a strong correlation between the presence and presumably biological activity of ERBB3 and the ability of cells to sustain proliferation. These cells to sustain proliferation, these cells that synthesize ERBB3 and activate it. And indeed, if you now interpose an animal model in which Joyce and Ching introduced into the peritoneal cavity of animals OV8 cells that had been armed stably with an inducible hairpin against ERBB3 in 2 different flavors, and now turn it on in the animal after proliferation had begun as measured by bioluminescence. Each one of these lines had been independently transfected stably with a luciferase allele. In this particular case, this hairpin led to a regression multiple times, and the animals were actually blinded. We didn't know the names of these animals. Well, you don't ever know their names. They carry little numbers around their chests. So these guys carried a number which wasn't 3293, and these guys carried a number that wasn't 3982. But in both cases, there was an anti-tumor effect. There was a survival advantage, a major survival advantage for 3293, a statistically significant and obvious one for 3982. And you can still ask yourself, maybe we just got lucky or unlucky, And these two hairpins have, as yet, undetected off-target effects leading to anti-tumor effects, which have nothing to do with their ability to compromise ERBB3 synthesis. So Joyce did, with Ching, another experiment, which is still afoot, and I'm pleased to report we now have the final answer. I'll show you the preliminary results of a few weeks ago. Didn't have time to make the slide. Joyce and Ching asked a simple question. Let's take OV5 and OV8 cells. Let's arm them with the relevant hairpins. And let's test them both in parallel. And most especially, let's pay attention to OV5 because OV5 cells produce ERBB3, but they don't activate it. So now if you deprive the cell of ERBB3, not having had an anti-proliferative effect in tissue culture, what happens in the animal? Answer. All right, first we did kinetics in the animal in tumor of the disappearance of ERBB3 over time. So this is like a PD effect in a lump in an animal over time after turning on DOX. 3293 depletes the protein, so does 3982, but this one depletes it considerably faster. Repeatedly. This is one experiment. This was done several times, always with the same result. And so then you can ask, all right, let's compare these guys in OV5 cells. And in two separate outings with the two different hairpins, compare the two colors, you can see tumor formation, again, as measured by luciferase increase, was no different between the two hairpins, and there was no anti-tumor effect in the case of OV5, in keeping with the notion that these hairpins are not the unfortunate survivors of a selection that was abiological. In short, these hairpins don't seem to deliver, so far, any indication of an off-target effect, and you can see that these OV5 animals die at about the same rates, whether or not they saw either hairpin on or off. And so far, we have no evidence in these experiments of an off-target effect. And I'm going to show you further that we have evidence that very strongly suggests that the effects you do see are on target. Now, Joyce decided to pursue the mechanism by which ERBB3 is activated in these cells a little further. Well, the first question she asked was whether or not the same cells that activate ERBB3 after its production produce the ligand. And the way she did the experiment was kind of neat at the time. She took tissue culture supernatant from OV5 and OV8, and she delivered it to OV5 cells. And when you deliver it to OV5 cells, in fact, if you put OV5 supernatant onto fresh OV5 cells, nothing happens. You don't see any activated ERBB3 or anything else. If you deliver the conditioned medium from OV8 cells to OV5 cells, now you see a band here, which it turns out is the neuregulin 1 beta subunit itself. And if you now take these tissue culture supernatants, put them on top of OV5 cells, here's OV8 tissue culture supernatant, here's a supernatant from another cell line in which we found activated ERBB3 called T21, here's synthetic neuregulin 1 beta, and here's the supernatant from OV5 cells itself. OV5 supernatant results in no activation of the ERBB3 that's in these cells. The positive control growth factor does, and so does supernatant from OV8 and T21G, as if some of these cell lines are actually producing the ligand that activates ERBB3 itself. So then Joyce went further, and she found in Western blot experiments that she could detect the secreted neuregulin except after its depletion with two different siRNAs directed against neuregulin 1 beta. And just to go a little faster, she did another experiment to ask whether the activity, the growth of OV8 cells was dependent on their ability to synthesize neuregulin. So in this case, she showed the naive cells that had been stably transfected with a vector, two different siRNAs directed against neuregulin, and in both cases obtained an anti-proliferative effect. But then one of these siRNAs, one of these siRNAs has such a sequence that a particular cDNA encoding neuregulin can be rendered immune to it by mutagenesis. So if you deliver that cDNA into these cells, you override the anti-proliferative effect partially, but reproducibly in 5 separate experiments. If you deliver the straight, unmodified cDNA that's not inured to the effect of the siRNA, in parallel, you get no override of the anti-proliferative effect, as if the effect of neuregulin is specific as well. So now we have a cell line like OV8 that produces neuregulin, that needs neuregulin to drive the proliferation of OV8 cells, in which one needs also activated ERBB3 to result in the proliferation of these cells. The scene was beginning to grow moderately interesting. And so Joyce did another experiment in which she asked, we've been studying proliferation, but that's only one of a myriad of characteristics of a tumor cell, about the ability of a tumor cell to aggregate with itself. And so it's been well known in the ovarian cancer field and many other solid tumor fields for years that primary epithelial tumor cells can sometimes self-aggregate. They form spheroid-like structures here. If you take OV8 cells and introduce those two different hairpins and deplete ERBB3, now you can show that the ability to self-aggregate is compromised significantly, just as the ability of the cells to proliferate is compromised. And aggregation, in the case of ovarian cancer, is probably an important early step in the natural human pathogenesis of the disease. And so then Joyce and Ching, in collaboration with colleagues at Merrimack, decided to do an experiment that we thought was important in this area to try to eliminate as much doubt as possible that ERBB3 might be interesting clinically and therapeutically. So what they did basically was to grow OV8 subcutaneously and obtain from Merrimack a monoclonal antibody MM121, which is directed against the ectodomain of ERBB3, and delivered to these animals versus a control in multiple experiments. Here's one of 4. And in each case, the ability of the cells to grow using calipers, an old method to measure lump formation, was significantly markedly reduced in the presence of the antibody compared to the control, as if there was something about ERBB3 that may control proliferation. It might control aggregation, but it's controlling lump formation, and that's what really counts in this particular kind of experiment. And so I think taken together, these experiments strongly suggest that the neuregulin 1 to ERBB3 pathway, because you remember these cells growing under the skin of an animal, they are human. They were once a part of a papillary serous ovarian cancer. They are autocrine. That is, the neuregulin ERBB3 circuit is operating in them. It's required for proliferation. It's presumably required for lump formation. And so I think one could argue fairly at this point that this autocrine pathway is a validated therapeutic target in papillary serous ovarian cancer growing in an emphasized preclinical setting. And so the question is it a real target in people? Which is the only thing that really counts. So let's talk about primary ovarian cancer cells for just a bit, and then I'll quit. So Joyce decided, having found that OV8 cells and a number of other established papillary serous ovarian cancer cell lines synthesize, secrete, and use their own neuregulin to drive their own ERBB3 to activity, she measured neuregulin-1 messenger RNA by RT-PCR in a collection of primary, non-established, non-cultured cells directly out of Dana-Farber Cancer Institute ovarian cancer patients with papillary serous disease, and found a signal in a number of them. And these primary cell packs, uncultured, merely washed and introduced into medium and freed of red cells and polymorphonucleoleucocytes, lymphocytes, and so forth by anti-EpCAM capture, were allowed to sit overnight. Their conditioned medium was taken and it was introduced onto OV5 cells, and you can see that some of them result in ERBB3 activation compared to a control, just like OV8 tissue culture supernate. That was stimulating at the time, and Joyce did that experiment a number of times. Here's a cleaner representation thereof, a more extensive one, where you move the tissue culture supernate from a primary culture, not even culture, primary strain, non-cultured cells from DF5, they're called, DF14, they're called. You get not only activation of ERBB3, you can see it co-precipitates with P85, as if in Mrs. Jones's cells, the actual pathway to PI3 activation is on. And then you can ask, okay, what happens if you show these same cells MM121? Answer, if you wait 3 or 4 days, ERBB3 activation disappears. The protein doesn't disappear, the state of activation disappears, as if there's something about this circuit that's operating even in a minimal laboratory setting. So then the question is, okay, can these cells be employed in animals to condition one to think about a clinical trial? And I can tell you that we now have 17 of these characters, and they all generate tumors. That's not new. It's been known that you can take primary ovarian cancer for years and generate transmissible or transplantable tumors. Here are examples of 4 of them. The tumor pathology is indistinguishable from that of humans. Occasionally, you even find malignant pleural effusions, just like in patients. Sometimes, rarely, in an occasional animal in which there's been transplantation of one of these primary strains, one can find even pulmonary metastases and hemorrhage. The cells themselves stain by traditional histopathology with antibodies directed against certain antigens that are known to characterize papillary serous disease. And we now have 17, and more are coming, we hope, of these primary guys. Most of them do not proliferate in tissue culture. Most of them are not viable in tissue culture, a major problem that needs to be overcome and that many people are working on. But of the 17 that are transmissible, 7 of them have the autocrine phenotype. They produce neuregulin. They synthesize ERBB3, it activates the PI3 pathway, and where we can test it, we can move the tissue culture supernatant onto OV5 or like iGrowth and get activation. So, something that was seen in the original experiments in established ovarian cancer cell lines is true in primary cells. So let's go a bit further. Indeed, in exploring these 17 primary strains, the same 4 flavors that we saw in the cell lines are there. About 30+% of a small number, 17, have the or express the autocrine phenotype. The autocrine phenotype is accompanied by neuregulin expression. We can find the precursor and the product proteins. We can show that you move the supernatant onto OV5 cells and see activation of ERBB3. And furthermore, if you take these primary cells and get a few of them to proliferate, albeit slowly in tissue culture, when they do proliferate, one can show that the same hairpins which block proliferation of established cell lines also compromise proliferation of these cells, even in an inducible state. So you can induce a hairpin in a primary cell. That's not easy because these cells are not easy to transfect, in this case, to infect with lentiviruses, but you can. We're now trying to introduce dominant negatives of ERBB3 in an effort to recapitulate what might be a therapeutic effect in the primary cells and try to develop better predictive biomarkers. One can show that MM121 in primary culture, albeit short-term, because as I pointed out, these cells don't grow very well, in this particular primary cell, will compromise ERBB3 activation and, to some degree, AKT activation. That's the monoclonal anti-ectodomain antibody. And so now the last set of questions we're asking at the present time is, okay, ERBB3's got a bunch of potential heterodimeric partners. Who among this very small set, and there are other proteins like MET, as shown by Jeff Engelman and Pasiani, that potentially could serve as pseudo-partners of ERBB3 or operative in these primary extracts. I'll skip this just by virtue of time, but let me tell you that the most abundant, the one ERBB family member that seems to be present in the majority of the primary strains that we've analyzed is ERBB4. That was a surprise because in the established cell lines, we rarely found it. And it is associated with ERBB3. Here's an anti-ERBB4 IP. You can see that anti-ERBB4, of course, brings down ERBB4. Anti-ERBB3 co-precipitates ERBB4, and so on, 4 different primary strains. And the ERBB4 protein is seemingly abundant. In fact, let's see if we can get this to go forward. We've screened now some of these, and you will note that in this cross sort of hatch analysis, ERBB4 seems to co-IP in the 6 lines we've tested it only with ERBB3. And ERBB3 seems to co-IP in a number of lines with ERBB4, as I showed you, and in the others with ERBB2. There are interactions between ERBB2 and ERBB4 in some of these lines, but the black means they're weak. I can't put a number on them, but they're hard to see. And EGF receptor is associated with ERBB3 as well, but these appear to be weak or minor interactions. And so what's the significance of the presence of ERBB4 in this setting? We don't know. But one observation I can tell you about that is reproducible where we see it in those 4 lines, we have hunted for any indication that ERBB4 seems to be affecting the ability of ERBB3 to be phosphorylated on tyrosine in 2 different sites. And the answer is when you knock down ERBB4, if anything, the degree of phosphorylation goes up. And the proliferation doesn't seem to go down. So is this some sort of interesting, nefarious player? We're going to have to find out probably by proteomics and some, hopefully, intelligent genetics. But it's clearly part of the story, and it does need to be understood. So finally, what else are we doing? And then I'm going to quit. So I think many of you know that elegant technology has been developed by Scott Lowe and Mike Heeman, Lowe at Cold Spring Harbor and Mike Heeman at MIT, that allows one to screen in lumps for genes of potential interest that may affect the tumorigenesis process. Those potential players that compromise tumorigenesis can be scored by deep sequencing or by array analysis if one has a very robust response. And ideally, these targets could be different in some cases from those you might pick up in a simple proliferation or even a viability screen done in tissue culture. And so Nellie Barata in the lab has set this system up. It hasn't been terribly easy, but she now has the pilots working. I think we can see a needle in a haystack now by deep sequencing. And so in reconstitution experiments, she can find ERBB3, she can find neuregulin. So that's reassuring because this is scoring directly in a lump of OV8 cells growing in the peritoneal cavity, and we'll see what else is found. These could be illuminating new targets, potentially worthy of investigation. Second, in tissue culture, we never see death when we knock down ERBB3. Even if you can't see the protein, there's no death. In the animal, there's death, but there's no death in tissue culture. That raises the interesting possibility that ERBB3 anti-proliferative effects could be more potent, and there could be associated apoptosis. And so Stoyle and Joyce Liu have set up a hairpin screen that's aimed at trying to potentiate, search for potential targets that potentiate the anti-proliferative effect of ERBB3. And finally, together with colleagues in industry, we are in the business of trying to set up the first of, hopefully, an interesting series of clinical experiments. I thank you for your time and attention.
Okay, questions for David?
Is that Mark Goldberg? Good to see you.
It's a great talk. Great talk. I was curious, is there a mutation that can tie all this together in this subset of patients that have this autocrine loop? Is question one. And the question two, kind of related to that, just thinking about those clinical trials, do you feel you know enough to screen patients who should enter those trials that show the phosphorylation?
Right. So first, we do know the following. So we have, I haven't had a chance to go through this, in the cell lines where there's an autocrine phenotype, and in the primaries where we think we have the makings of an autocrine phenotype, we scored for amplifications of neuregulin, don't find it. We haven't deep sequenced the neuregulin allele. I'd be surprised if that was a winner. We have, however, sequenced the ERBB3 alleles in a number of them, maybe 25% so far, found nothing. I'd be surprised at that, but it's possible. So far, I haven't found it. There's more to go. I mean, we need to examine the genomics, genetics, and functional behavior of ERBB4, because there could be other guys playing an important role in this that have a down modulatory effect on the pro-tumor development activity of ERBB3. That may be, if you allow it to go on, kind of uninhibited, eventually the outcome could be lethality. That's not good for a tumor cell. We need to find out. And finally, Mark, MM121 is in clinical trials in the United States and Dana-Farber as well.
Are they selecting patients based on?
Not yet. We do need a valid predictive biomarker, which is, I think, kind of obvious, but it's unfortunately a major need throughout the field, as you know better than most. Yeah, Jose.
This is fascinating. What I find really interesting is this ERBB4, ERBB3 co-precipitation, because in the field of ovarian carcinoma, ERBB3 had been targeted thinking that the dominant partner was going to be ERBB2. the clinical trials with pertuzumab that basically is blocking the ERB2, ERB3 dimerization showed some activity, but nothing exciting, and maybe
Lapatinib was dead in these models.
Yeah, so maybe that's what it is, you know. So this is so the idea of hitting ERB3 directly, if anything, makes, from what you said today, even more sense in ovarian carcinoma than in breast cancer, because breast cancer is much more an ERB2-driven condition. So this is very interesting. Now, the cell lines that express. So is there a correlation between ERB4 expression or co-precipitation and sensitivity to these antibodies? Because that would be a very obvious biomarker to look at.
I would say it's coming to your theater someday if we could ever earn a ticket price. I don't know. We're working on it. It's too early to say.
Robert?
Real important question.
Over here.
Maybe you said that and I just overheard. So is ERB3 overexpressed in ovarian tumors compared to healthy ovarian tissue? Or you haven't done these patient tumor experiments yet?
There are actually papers in the literature from others that demonstrated that in a significant fraction of papillary serous ovarian cancer, ERB3 is overexpressed. What that means, I can't say. In some of these cell lines, the abundance of ERB3 is relatively low, but you know, presumably a lot of it's hard to know whether a lot is Western blots. It's hard to know quantitation, but the impression is that a lot of it is activated, and those cells are totally dependent on a small amount of ERB3 by comparison with OV5 cells that produce, you know, n-fold more and aren't dependent at all. So I'm not sure what overproduction means.
So roughly what percentage of ovarian cancer patients could benefit from ERB3?
Well, we found from a small set, emphasize small set, these are small numbers, 17, that over a third reveal the autocrine phenotype, as defined by not just the presence of messenger RNA for neuregulin, the protein itself that when moved on to OV5 says has biological activity, the presence of an activated ERB3 polypeptide, at least at one site, and its ability to interact with P85 in the same non-clonal mixed primary Mrs. Jones-derived cells.
David, the heterodimerization is clearly going to be very important here. The ERB4 component of this looks like it could also be equally important in terms of targeting. So have you any thoughts on that?
Well, Patty, I mean, thoughts aren't worth much these days. They go down faster than Democrats, actually. The Democrats,
well,
I hope,
spoken
don't.
on today, too.
I don't care if they're Republicans or what, but Democrats are going down today. That thought could go down, too. So I don't know. It's too early to say. But the one thing I can say is in the two cell lines, not strains yet, we see the cooperation, we see heterodimerization in the strains. In one cell line, we could find ERB4, and in one cell strain, knocking down ERB4 didn't seem to compromise viability yet in a short-term experiment.
David, before Bruce demands equal time for the Democrats.
In this town, the Democrats get more than equal time.
Well, in that
About
case,
9-1.
In that case, Bruce, you first, and I'll ask mine second.
Well, for purposes of disclosure, David's a Democrat, so don't take it personally, anybody.
My wife's a Republican, by the way.
Oh, is she? Oh,
she
God,
is.
no. That's right. There's a little history there.
Yeah, there is.
So, David, you never get rid of proliferation entirely by knocking ERB3 out. Is that true? It seemed like some of the cell lines there was just less proliferation.
That may be a result of not being able to deprive the cell of enough ERB3 by RNAi. Yeah. It may not necessarily be. Yeah. In an RNAi experiment, it's always difficult to know. Just because you can't see the protein doesn't mean it's not active, especially if it's tied up with an enzyme with a high turnover number. So I don't. But we don't see death, and we do see death in the animal. That's the key thing.
One of the possibilities is that you have a primary signal that's driving through ERB3, but also through other pathways. And I was thinking of other receptors that potentially could signal in multiple different pathways. Have you done a screen for other active kinases?
Well, rather than doing that, what we thought we would do is do this screen in the animal in a lump of a human xenograft, because from the therapeutics point of view, that's getting us close to what might be a reasonable target in a real, albeit preclinical, clinical-like setting. Also, in this potentiation screen, we might find other, right? Because that potentiation screen would be done with druggable, with the hairpins that are directed against druggable proteins, like kinase. Absolutely, I think there's a lot more than meets the eye here. Probably 100-fold more than meets the eye than you can see. I'm with you.
Marty.
Thanks, Patty. David, this has been an encyclopedic overview of a tremendous amount of work, and it occurred to me that perhaps a word maybe from your colleagues at Merrimack might be helpful, just to describe for us a little bit about MM121 and its derivation, as it certainly is playing a very important role. Maybe Clet or someone? Here. Bill.
Thank you. Well, I haven't prepared anything, but it's been a great collaboration with Dr. Livingston and Joyce and the rest of the team. And as Dr. Livingston mentioned, we're entering clinical studies with MM121 in collaboration with our colleagues at Sanofi. And we are looking at markers. We're very committed to trying to find biomarkers for MM121. And so we want to make sure, for example, that they're not prognostic versus predictive. And so our first foray into this is going to focus more on taking all comers and trying to sort out who's responding and who's not responding, that, and correlate that with heregulin or ERBB3. And we have a set of biomarkers in which we plug them into an algorithm to try and predict these kinds of things as well. But right now we are in the clinic and just entering in ovarian cancer with Taxol, in combination with Taxol, and we're working with Joyce and the group to get biopsies as well. We're also in a non-small cell lung cancer study in combination with erlotinib, and a breast cancer study in combination with exemestane. So if any of you have patients that you want to enroll.
Okay, well, thanks very much, David, for a wonderful expose.